Sesim bağladım.
Can you hear me?
Hi everyone, can you hear me?
Yes.
Okay. Hi everyone, greetings from Baku studio. It's honor for me to introduce Robert Langer. I will keep in short. Robert Langer is professor at the Massachusetts Institute of Technology. Professor Langer develops long-term and controlled-release drug delivery systems for a variety of applications, including cancer therapy, insulin, and vaccines. Dr. Langer has received over 220 major awards. One of them is 2014 Breakthrough Prize in Life Science, which recognizes excellence in research aimed at curing intractable disease and extending human life. It's your floor, Professor.
Thank you so much. So if we could turn the slides on. Thank you. So anyhow, yeah, that's okay. We can start on that. What I wanted to say is, I thought I'd take you through some of what we've done in my career, which didn't start out all that great. But anyhow, I got my PhD in 1974. And when I did, most of my friends went into the oil industry, but I wasn't that excited about it. And I received a job offer actually to be a postdoc, which was by far the lowest paying job, but I was excited about it, in a surgery lab at Boston Children's Hospital. And I'm a chemical engineer, but I was the only chemical engineer or the only engineer in the entire hospital. Dr. Folkman had a very interesting idea. If we could have the next slide. His idea was that if you could stop blood vessels, that might be a whole new way to stop cancer. It's kind of depicted on this slide that tumors start out on the lower left as they're somehow abnormal. They grow in a three-dimensional mass if you follow those arrows. If you get to the middle lower part of the screen, maybe you have about 1 million cells, and the cells in the center die. You reach an equilibrium. It doesn't get to be more than that because of a nutrition problem. The cells in the center die because they can't get nutrients or get rid of waste. But the tumor solves that problem because it secretes a signal, he said, called TAF, or tumor angiogenesis factor, that causes surrounding capillaries and blood vessels to grow right to the tumor. So if you follow the arrows again on the upper right, you see the tumor getting bigger and bigger because now it can get nutrition from the blood vessels. And when you go to the uppermost right, what you can see is that the tumor is highly vascularized. And what happens is the tumor spreads through those blood vessels. That's a process called metastasis. And that's what eventually kills. Dr. Folkman's idea was that if you could stop blood vessels, maybe it would be a new way to stop cancer. People didn't believe him. Anyhow, he hired me to try to prove he was right. And in so doing, isolate the first blood vessel inhibitors. Critical to that problem was creating what we call a bioassay. If we could go to the next slide. And to create a bioassay to study blood vessels, one didn't exist. And so we put certain types of tumors in eyes of rabbits, and then delivery systems, which could be polymers or lipids, right next to it to deliver large molecules. That's what we were trying to isolate. The only thing is that when we talk to people who are really expert in this field, and there weren't very many at this time, 1974, they all told us this wasn't possible. They said, you can't have next slide, you can't have a delivery system that could release large molecules because they're just too big to diffuse through solid materials. Just like none of us could walk through a brick wall, we're too big. Nonetheless, if we were going to solve this problem, we had the angiogenesis problem, we had to solve the delivery problem. So I spent 2 years in the laboratory experimenting with different techniques to make microparticles and nanoparticles, pellets, and over 200, I found over 200 ways to get this to not work. But eventually, next slide, I got one, I was able to make little micro or nanoparticles, and we published in Nature, next slide, in 1976, that for the first time, you could release large molecules like proteins. And even this is the first report of, to my knowledge, of anybody ever releasing nucleic acids like mRNA or DNA from tiny particles. Now, a couple months before this paper came out, I remember giving a talk at a major scientific meeting in materials. And when I got done, I was 27. I thought, the older scientists in the room, they would want to encourage me, this young guy. But when I got done, a whole bunch of them came up to me and they said, "We don't believe anything you just said." They said, "It's just, if we could go to the next slide." They said, "You just can't release large molecules. They're too big," as we heard before. But also, I was using organic solvents like ethanol and methylene chloride. And they said that would destroy whatever we put in of these sensitive molecules. And actually, things went downhill from there. I got my first 9 grants rejected because people kept saying this was impossible, that you couldn't do this. I tried to get a faculty position in a chemical engineering department, but not a single chemical engineering department would offer me a job. They said this biology stuff I'm doing doesn't make any sense for an engineer to do. I finally got a job in a nutrition department. I received it, but because the man who was in charge of it, Nevin Scrimshaw, very famous nutritionist, liked me, and he offered me the job. But he didn't bother to ask anybody else what they thought. So that might have been okay, except for one thing, which was the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and they said I should leave too. In fact, if we go to the next slide, what people said is that this is Mike Marletto, one of my good friends, reflecting back in the late 1970s. And what he said was, one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientists heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, "You better start looking for another job." But you have kind of two choices, right? You could keep trying or you could give up. And I kept trying because I believed in what I was doing. And like I said, we wanted to solve the angiogenesis problem. That was the highest priority. And I mentioned that eye assay we were using. So now I'm going to show you a slide, and it's kind of graphic. So some people may not want to look. It's bloody. But there's no other way you can show stopping blood vessels. So what we did, if we could now go to that slide, the next slide, what you see, this was published in Science, the journal, in 1976. Without the inhibitor on the left-hand side, and we did this experiment over 1,000 times. Every single time you get this sheet of blood vessels. We also, I always say we had a lot of controls because almost everything I isolated and we put in didn't work. But on the right-hand side, we found one that did. And this proved for the first time that you could stop blood vessels and to actually stop cancer by doing so. We repeated that experiment 20 times. And like I said, this was published in Science in 1976, but it still took another 28 years from our paper before the first blood vessel inhibitor was approved by FDA. Really, it took billions of dollars by companies like Genentech and others to using techniques that we and others developed. But starting in 2004, 28 years after this, next slide, what you see is one after the next blood vessel inhibitor is approved by FDA. Avastin, which is the first of these, that's probably the number 2 or 3 best-selling biotech drug in history. And there are many, many ways to now use these to treat all kinds of cancer and also eye diseases like macular degeneration. Another thing that Dr. Folkman and I talked about was filing a patent. Boston Children's Hospital at that time never had a patent. So we filed for a patent for the first time. And 5 years in a row, the patent examiner rejected it. And everyone told me again I should give up because he wasn't going to change this. But I don't like to give up. So I started to think, how could we convince the examiner to allow it, legally, of course. And science really wasn't working. So, but as I told you, when we first started doing this, everyone said it was impossible. It could never work. I wondered, maybe someone wrote that down. So I did this science citation search in 1982, 6 years after our Nature paper came out. And I found very interesting papers. If we could go to the next slide. This was from 5 of the top material scientists in the world. Let me read you what they wrote. And I had no idea this was written, but they said generally the agent to be released, they're describing the field, is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising is a really good word for a patent examiner, that clearly demonstrate to the opposite. So I showed that to the examiner, and he said, gee, he did no idea, but he'll allow the patent if I could get written affidavits from each of those 5 authors that they really wrote it. So I was this young guy and I wrote them, but they're all nice enough to write me back that they really wrote it. And we got this really, really broad patent. Next slide. And we use that patent to start companies, to teach companies how to do it. And because of that, and these principles that we develop over the years, now there are many, many products based on this. The next slide just shows you pictures. I won't go over all of them, but what you see in the middle of the screen is Lupron Depot. This was developed by Takeda, who we had a relationship with. And they're little microspheres that can release luteinizing hormone, releasing hormone analogs of peptide. And they're the most widely used treatment today for advanced prostate cancer. There are many, many others, treatments for opioid addiction, schizophrenia, heart disease, eye diseases, and so forth. They're used by tens, if not hundreds of millions of patients around the world every year. But you always like to do better. And we had made microparticles and nanoparticles, but sometimes nanoparticles would stick together. Sometimes they would be eaten by macrophages. So what we did, next slide, is we came up with a coating that we could put on these with polyethylene glycol. This was published in Science in 1994, almost 30 years ago. Let me just show you a video about how it works. Next slide. Can we play the video? Is it not working? I guess it's not. I thought that they were checking the video before. Maybe nobody checked it. Well, why don't we go on? I thought this would have been checked. I thought my office had told people to check it. Can we go to the next slide? So anyhow, I'm sorry about that. We had sent this talk a little while ago, and I thought that was going to be looked at. It's actually a pretty nice video, kind of explains how nanoparticles work. But anyhow, let me give you a real life example of what we have done with nanoparticles. So in 2010, myself and 3 other people, Nubar Fayan, Ken Chen, and Derek Rossi, had this idea to create a company to use nanoparticles and other ways to deliver what's called messenger RNA. Protein therapeutics had been very successful, but proteins take a long time to make, and there's a lot of diseases they can't treat. RNA could make the protein if you could deliver it and protect it. And you wouldn't even need to, it wouldn't take very long, and it could be very effective. Also, if we go to the next slide, you can even treat diseases you could never treat before, like intracellular diseases and membrane-bound diseases. So we started this company, Moderna, and we started it in 2010. Let me just show you some of the things that Moderna is doing on the next 3 slides. So this is the Moderna pipeline. Again, I'll go through this quickly, but they're using it to treat all kinds of COVID treatments. The next slide shows some other vaccines that we're treating, and the 3rd slide in this series shows some different diseases like heart diseases and cystic fibrosis and rare diseases and cancer that we're using. So Moderna, which we started in 2010, now has something like 3,000 people. But I thought I'd give you a specific example of what we did and show you the power of messenger RNA. The next slide shows what happened with COVID. Go back one, please. Back. Yeah, thank you. So in January 11th, you may know the Chinese scientists published the virus genetic sequence. In less than 2 days, the Moderna scientists made the design, put it in nanoparticles, and then within a month, it was shipped to National Institutes of Health for testing. And within the next month, people were actually injected. It was like I say, just about 2 months from starting this to that. It was really unprecedented. But just like I would receive a lot of criticism when I started, Moderna did too. So just to take 2 months later, I'll go now to May 2020, when we published or talked about the first clinical data. Boston Globe, if we could go to the next slide, which is our local paper, wrote a big article because they talked to all these so-called experts, and they basically said what we're doing. They put my picture on the cover. They said, this isn't how you do science. That was May of 2020. Just 6 months later, I remember being in our boardroom, and we broke the code for what's called the phase 3 clinical trial, the final trial before you get it approved. So there were 30,000 patients in that trial. 15,000 got placebos, but 15,000 got the vaccine. And what was remarkable is not a single person died, not a single person went to the hospital. And the vaccine was 94 to 95% effective. For reference, flu vaccines are generally, which take a year to make, are generally less than 50% effective. The next slide just shows a picture of it. And the next slide, I'm going to start showing you some data. Next slide. So basically, what you can see on this slide is the tremendous difference in hospitalizations when you get the vaccine compared to when you don't. And with a booster, it's even better. And the next slide shows you data from the Mayo Clinic that the Moderna vaccine, certainly in the United States, has been the most effective vaccine there is. 94% effective compared to Pfizer-BioNTech, which is 91%, and Johnson & Johnson, which was 60%, but removed from the US market because of safety concerns. If you look at other ones, they might be on the order of 50% when they really do it. And I'll just show you a few publications that back this up. The next slide show from the New England Journal with almost 400,000 people show, that this has the lowest rate of infection or hospitalizations, which is mRNA 1273. The next slide is another clinical trial or clinical data of over 400,000 US veterans. And again, mRNA 1273 was by far the most effective. Let me end by telling you where I hope this will go. And this is work that we're doing in our own lab at MIT. So Bill Gates, next slide, came to see me one day. And that's me on the left and him on the right. And he wanted to see if we could come up with a way to help people in the developing world where people don't come back for a second injection. And so the next slide shows what we were trying to achieve. We wanted this pulse delivery. So the idea is, could you someday give one injection and then have a cocktail of little particles that could deliver the drug, say at 1 month, 2 months, 5 months, a year? So what Anna Jakulenik and Kevin McHugh in our lab did is develop a new printing process where we could make little particles, but by changing the nature of the shell, they would break at different times. Let me just show you a video that covers what we did.
The stamped assembly of polymer layers, or seal method, combines manufacturing technology with a novel layer-by-layer assembling method to create an array of core shell polymer particles. First, the polymer of choice, PLGA in this example, is melt-pressed using a prefabricated silicone mold. The mold with the particles is then transferred to another substrate, where it is peeled off, leaving behind an array of polymer-based particles. These are then filled with any drug or vaccine using an inkjet piezoelectric nozzle and then dried. To seal the filled particles, a cap mold is aligned, sealed with the base, and delaminated. The resulting array of core shell particles are then removed from the base and stored until use.
So just to show, so again, please go back a slide. So these are just some pictures of the mold. And the next slide shows our paper in Science. This is a whole new printing process. Next slide. And we also have a patent where Bill Gates is an inventor. Next slide. But what's really exciting, I think, is that you can do just what we said. You can take this as in vivo data, and by changing the nature of the shell, like the polymer composition, what you can see is you can get bursts of vaccine up to 6 months, but you could do it really at any time. So the idea is you could give one injection with all these in it, and you get multiple boosters. We'd call this a self-boosting vaccine. And I thought I'd just show on a last slide, again, where we're going, where we hope this will go. And I'll make this the last slide. But what we're doing is working with one of my former students, Mark Prousnitz, who's at Georgia Tech, to put these in what we call microneedle patches. And you see the microneedles on the right. They really don't hurt you. They don't go very deep, but they can deliver the vaccine. And you see a picture of it on the left. It looks like a Band-Aid. And they basically, the idea is we could hopefully someday ship these all over the world. Right now, they're in animal trials, have patients use them and get multiple doses of vaccine, but with a single Band-Aid application. So let me stop there and see if there are any questions. And it's certainly been a pleasure to speak to you. Are there any questions?
Thank you, Professor, for the interesting lectures. Our students have a few questions.
Sure.
The first question is, okay.
Hi, everyone. Can you hear me?
Yes.
I'm glad to meet you. I want to ask some question. Isn't CRISPR-Cas9 an appropriate method for the treatment of cancer cells or genetic disease and etc.? Because it has a feature like cutting certain parts of the genes. And is it possible to adapt this method to non-genetic disease in the same way? Because in this case, there's no need for any vaccines, maybe.
Well, for cancer, I think it's too early to know. I think that CRISPR-Cas9 is certainly a promising technology, but I don't know that we know enough about all types of cancers to know whether that could be a cure. I mean, you'd need to know what to cut and paste, so to speak. And I'm not sure that I don't know that we know that. By the way, you also would need to know how to deliver it so that you could use it. So I think CRISPR-Cas9 is incredibly promising technology, but curing cancer, a lot of people have tried, and it's not a very easy thing, both because the biology is not fully understood, or they're not well understood, and the delivery approaches are very, very hard to do. I mean, but we've been working on that as well, but it's not an easy problem to solve.
Okay, I got it. And my next question is, in the treatment of cancer or any genetic disease, instead of placing microparticles in cancer cells, can they be placed directly in the human body to prevent disease or genetic disease?
I don't think that's so straightforward either. I mean, to me, again, the problem is, how do you deliver molecules that are going to prevent genetic disease? I think that all these techniques, I mean, mRNA, I think, has been incredibly promising, as we've seen with COVID. CRISPR, is in early stage clinical trials, and I think it's a very exciting technology. But, curing these diseases is a very, very tough problem. People have been trying for a long time. And like I said, I think we need to understand the science better.
Thank you.
Thank you, Professor. The next question.
Yes.
Hi, everyone. Can you hear me?
Yes,
Okay.
I do.
My question is, what can be done to prevent genetic diseases, also cancer, from being passed from parent to child? Or are there already ways to do it?
I don't think there are ways to do it. I do feel that vaccines could be helpful, that various types of genetic approaches could be helpful, but I don't think it's an easy problem to solve, but it's a very good question.
Thank you, Professor. The next question is from another student.
Hi, everyone. Can you hear me?
Yes.
We know that there are many vaccines now, so why are the side effects of the Pfizer vaccine greater?
I'm sorry, I didn't exactly understand the question.
We know that there are many vaccines now, so why are the side effects of the Pfizer vaccine greater?
Well, I don't know that the side effects of the Pfizer vaccine are greater. I think, to me, from what I've read, they seem pretty mild, compared to others, and Pfizer's, published the data. I think both the Pfizer and Moderna vaccines have been very effective and very safe. I mean, the number of incidents of problems have been, I would think certainly at least as low as flu vaccines or other vaccines.
Thank you, Professor. Also, we have one more question.
Sure.
Currently, many injections are used in the treatment of COVID-19 and also cancer disease or genetic disease. Most of it is given to injections. Is it more appropriate to replace with pills?
We're working on that. We're working on pills that could deliver large molecules. We've published now some papers for delivering insulin and even RNA. Using certain types of pills with a needle in it that you could inject, where the needle is made out of the RNA or out of the insulin. So I think someday that may be possible. But, again, it takes a lot of clinical trials to get it to be possible.
Thank you. Thank you, Professor. Our last question, will be my question. As I understood that for nanoparticles, we use polymer shell, yes, for delivery drugs. And my question is, this polymer shell can induce hypersensitivity or allergy reaction?
We really haven't seen that. I mean, we've used both lipids and polymers, and neither one have caused reactions. I mean, I would say an awful lot of these way before the COVID vaccine have been approved by the FDA and other regulatory authorities for all for all kinds of things. Like PLGA is approved for sutures, bone plates, other drug delivery systems, like that picture I showed you of all the different vials. So it's been tested on millions, if not billions of people.
Okay, thank you very much. Thank you for your inspiring and interesting lecture. Thank you for answering our questions. And thank you very much.
Thank you. Thank you for having me.
Thank you. Bye.
Bye-bye.